This is Mrs. Alexander, and today we're going to talk about how amino acids behave. This information is your front load lesson for activity 323. We're going to first start off with a little bit of review. For the end of course test for Project Lead the Way, Biomed, you do need to know what the structure of an amino acid looks like. Take a look at the bottom right hand corner. That shows what a molecular structure of a protein looks like, also called an amino acid. There's a central carbon in the middle. To the right, there's carbon and oxygen bonded. That's our carboxyl group. To the left, there's a nitrogen with hydrogen attached, and that is our amino group. There are several other hydrogens around the amino acid as well. And then at the bottom, there's a big box that says R. That R varies. It's called a variable group because it depends on what type of amino acid it is. Valine, cysteine, serine, tryptophan, depending on the amino acid will depend on the variable group. What you really need to know about amino acids is when they're formed and they're linked together to form polypeptide bonds, you have to use a dehydration synthesis. Dehydration synthesis can link any of those 20 amino acids together, and it does so by removing two H's and an O. H2O is the byproduct, that's water, and when it does this, it will link the peptide bonds. Notice the figure on the right. Once two peptide bonds are linked, they can continue to link until they reach a stop codon in the RNA sequence. There are four ways that the R groups can interact with each other. Those four ways are van der Waals forces, electrostatic charges, SS bonds, and hydrogen bonding. Specifically, the one having to do with water is hydrogen bonding, and that's the one we're going to elaborate on in this presentation. You do need to know the other three and be able to identify them in a multiple choice style question. You just need to know a small amount of information, just kind of in essence what they are. So the next slide is van der Waals forces. This occurs whenever opposites attract. So whenever those R groups and the different electrons come into contact, if there's a positive charge and a positive charge, they will repel each other, kind of like two magnets with the same charge. However, if you flip one over and you have a positive and a negative, those forces will attract. So when you think van der Waals, just think of opposites attract. Next, we have electrostatic charge. Electrostatic charge you've probably been a victim of in the past, and that's when you get up out of your chair and you shock yourself. Think of electrostatic charge when it comes to that. Really, for electrostatic charge in amino acids, all you need to know is that some amino acids have a positive charge, others have a negative, and then, while there is also a third type called neutral. Neutral, in essence, doesn't just mean no charge. It means that the charges are neutralized. So the amino acid has a positive and a negative pole. So again, electrostatic charge is when opposites attract. This can cause the chain of amino acids to fold. SS bonds. SS bonds refers to the amino acids that contain sulfur. Sulfur is an element on the periodic table. You might have smelled sulfur before. It has a really distinct stinky smell, almost like rotten eggs. And this element sulfur can be found on several amino acids, and those amino acids will seek out other sulfur-containing bonds. So SS just stands for sulfur-sulfur bonds. So just know that the S, what it stands for, it's the amino acids that have sulfur, and they will look and try to form covalent bonds with one another. The last of the four ways that amino acids will react with each other is through hydrogen bonding. We've talked about hydrogen bonding several times this year, and specifically how it has to do with water. When it has to do with water, we usually describe this as polarity. Polar, meaning it has a positive and a negative poles on the molecule, or it is a neutral charged molecule, and nonpolar, meaning it's not charged neutrally. It's either positive or negative. So we can break those into two groups. Polar, meaning it has a positive and a negative. Nonpolar, meaning it's either negative or positive. When it comes to water, water loves to dissolve many substances, and water is considered a polar substance, whereas oil is considered a nonpolar, or it repels water. So remember those kind of tips when you're trying to remember how hydrogen bonding occurs. Whenever something loves or seeks out water, we call that philic, hydrophilic. However, when something rejects or repels water, we call that hydrophobic. So let's talk about hydrophilic first, water-loving. Neutral molecules that have a positive and a negative charge on them or are neutralized out will seek out water molecules. They love to dissolve and mix in with them. So whenever we call water polar, we can also refer to that as water-loving or hydrophilic. This causes the amino acids to stretch out in a big chain. Here's a picture of an amino acid chain. Notice it's not folded, it's just stretched out. The biology perspective on this is that amino acids that are polar or water-loving, they want as much as their surface area volume to touch water as possible. Think of water-loving or hydrophilic as stretching your arms out in a big, like I want to hug you motion. And what you're doing is you're really stretching your arms out and allowing as much of your arms as possible to touch the surrounding air. Well, in this case, it would be water if we're talking about polarity. Glutamic acid is a good example of a molecule that's polar. And if I were to run this simulation, which I will do tomorrow for you guys in class or sometime this unit, you would see this molecule moving around, wiggling around, and the blue background, which represents water, would be touching all the glutamic acid chain, and it would be lots of surface area to volume. Unlike valine, which is a amino acid who is hydrophobic, hydrophobic or phobia refers to fearing or repelling water. And so whenever somebody is phobic of something, like arachnophobia, afraid of spiders, in this case, hydrophobic is afraid of or fearing water. The most common substance you can think about is oil. Oil is hydrophobic. Whenever you mix oil and water together, they do not mix very well. But in this case, amino acids who have a hydrophobic polarity will repel water. They will avoid these chains, and instead they will herd or bunch together and fold into a shape. Here's a picture of that folding occurring. The picture on the left is a chain, and on the right is as it's folded or it clumps together. So instead of imagining yourself with your arms out ready for a big old open hug because you love water, think about pulling your hands in and cringing because that person you really don't like, or in this case hydrophobicity, meaning afraid of water, just trying to bunch up and keep as much of your body from touching the air as possible, or water in this picture. So bunched up molecules will fold in whenever they're afraid of water. How this amino acid property affects sickle cell anemia and normal blood is that normal blood is usually a nice circular shape. This is because it loves water, it has lots of surface area to volume. But the moment we change the glutamic acid to valine, that single point mutation, or that substitution from an adenine to a thymine, we get this bunching or folding. And that folding causes the sickle shape, or causes one part of the normal hemoglobin molecule to fold in and pinch in that sickle shape. And our last slide here just talks about oil and water, which we mentioned earlier. Since water is hydrophobic, that means it hates water, so what it will do, it will actually dissolve in other hydrophobic molecules. So oil dissolves well in other oil, and they do not like water. This is the end of your front load for 323. If you have any questions in class, just give me a holler.